Well, thank you. Good morning. So the project I want to talk about today has to do with the effects of an environmental pollutant on human health. And it affects the molecular level, the physiological level, and so on. So we are talking about multi-scale systems, and I want to show you an approach that we adapted to be useful for multi-scale analysis and exemplify it with this project I mentioned. And so this is called a template and anchor approach. So you're all aware that biology happens at different scales, and the scales are in terms of time. It goes from milliseconds to years, and sort of corresponding to that, the spatial scales that go from nanometers to many thousands of kilometers. And if you look at what fits into there, there's sort of an almost monotonic trend that shows you it goes from small molecules to ecosystems. And in many cases, these two, the scales are represented in the same model. For instance, if we look at this cyanobacteria system, they are very, very small, of course, but they account for 20 to 30% of the Earth's photosynthetic activity and recycle about 40% of the entire carbon dioxide. So we have a huge system in the ocean that is driven by very, very, very small organisms. And the question is, how can we deal with this multi-scale nature? Now, in some cases, we need to use nested models. So we have a model and we look at a detail, and if we blow it up, then there's another model, and if we blow it up, we have another model. So I said, how can we capture that? Now, in some cases, there is the well-known method of timescale separation, and I'm not going to discuss it today because I don't have time, but I'll be happy to talk with you during the break. But if the timescales are close, you cannot separate them, so you have to deal with them in some manner. So what we are proposing is to use the template and anchor model approach. And so the template models are high-level. It's a high-level, coarse-grained model that contains just the coarse picture of what's happening in that system. The variables in there are actually anchor models. So the anchor models, I'll talk about those in a second. They deal with the details. So we have the big model, and then we have small models underlying. The template models usually are running on a slower time scale, and they could be ODEs, they could be discrete models, agent-based models, whatever fits the context you are working in. Then we have the anchor models. So these are the fine-grained models that describe the detail, whether it's metabolism or whether it's some signaling or physiological aspects. And they could be any format too. They are self-contained, and they are connectable within and across the different hierarchical scales. And they typically run on a faster time scale. And as a computer scientist might say, they can be hot swapped. That means you can take them out, replace them with another anchor model while the rest of the model is sort of running. Now, I need to say the people who came up with these, Full and Karliczek in 1999, they used this template and anchor model approach for for a rather different purpose. Namely, they studied locomotion, and they said there must be some design principles that every terrestrial locomotion has to satisfy. And then whether you have a cockroach or giraffe, they have to satisfy this, but then there are all these different implementations. So they considered the design principle of locomotion as a template, and then the specific implementations as the anchor models. So we do it differently here. We use this approach to subdivide big multi-scale model problems into several small ones. So what are the features and the advantages of this type of approach? Well, the first one is it's a true alternative to other multi-scale approaches. There are many multi-scale approaches. And in a sense that we do not attempt to capture everything simultaneously. instead, we want to analyze the anchor models sort of beforehand and then just retain the important information, input-output information from these anchor models to calibrate the template model. The template anchor approach provides a clear structure for setting up these models, and that's important. A little later I'll show you that, and they're very flexible for moving across different levels. So the application is a health risk assessment for dioxin. Dioxin is what's called a POP, a persistent organic pollutant. It comes from industrial processes, primarily from incineration, but there have been bad exposures from Agent Orange in the Vietnam War. In Soweto and in Italy, there was an industrial accident, and there was something with food supply chain in Ireland some while ago. And in that graph, you don't need to see the details, but you can see that's an accumulation of dioxin in the human body over age. Okay, so it starts here with 20 and goes up to about 80. And you can see it's going up. Why is it going up? Because it can be in the fatty tissue of a human for many, many years, for decades. So it's very persistent. So here is sort of our template model. So we have the liver, and we want to study cholesterol because cholesterol is affected by dioxin, and cholesterol is produced in the liver. It goes into the intestines, it goes to the bloodstream, of course, and then it has to do with the reproductive system. So cholesterol is responsible ultimately for estrogen and testosterone and so on. And then it's in peripheral tissues because cholesterol is a very, very important. It's always maligned. Don't eat cholesterol, it's bad, bad, bad. No, you need cholesterol because it's in every single cell membrane. So it's an important compound of life. So we want to focus primarily on the liver, and that's an example of an anchor. Others we are working on, we have not done totally. So we want to look at the liver, and within the liver, there's this pathway that produces cholesterol, and I know you cannot see the details, but the details are not so important. It's just, there is much behind this. It's not just words. So it's a pathway that produces cholesterol, and there is an interesting discrepancy, apparent discrepancy, namely if you have this exposure to TCDD, dioxin, then in many cases, if it's bad enough, you get non-alcoholic fatty liver disease. But if you look at the molecular level, the activity of this biosynthesis pathway is reduced. All these genes are reduced. The enzyme activities are reduced. So how can it be reduced? And then you have too much fat in your liver. So we want to study that. And so we set up a model. And it was primarily a mass action model with some power law ingredients for regulation. And we calibrate that with literature information. There's a lot known about cholesterol, of course. And then we determined the effect of dioxin on the molecular processes, on gene expression. And that is known to some degree. And then we determined dose-response relationships between the amount of dioxin that goes into that body and the effect it has on these molecular components. So it's a dose-response curve that we get out of the thing. And we use that later in our template model. We did the analysis twice, once just by looking at the mevalonate pathway, and once with the mevalonate pathway, but also exchange between the liver and plasma. And there are many results. Here's the main result. If we start with the blue one, that's the hepatic cholesterol with mevalonate only, and you can see with increasing dioxin it goes down. Why is that? Well, because the activities, the gene expression and the activities of the enzymes are reduced, so production goes down, concentration goes down. However, if you look at the reddish curve, it goes up. And that's the same model, but it includes the relationship between the liver and the plasma, because there are all these proteins in the plasma that transport cholesterol, and there is a change by dioxin in how much goes into the liver, comes out of the liver. So you can see that's going up. and it's going up in a sort of monotonic fashion. It's not the S-shaped curve that we usually see in these types of exposures, but it goes straight up if you get dioxin exposure. Other results are, so we can look at other different subsystems, for instance, the reproductive system. We can look at the plasma itself, we can look at the peripheral tissues, and so on. We can check whether it makes a difference whether you're a high-level cholesterol diet or low-level cholesterol diet, whether those two things interact, and to some degree they do. We can look at subgroups. So for instance, different genders, different ages, races, and so on. And if we had we had the data on individuals, on enzyme activities and exchange between plasma and liver, we could do a personalized assessment of the risks that people have by being exposed to certain concentrations of dioxin. So we could do an overall health risk assessment for different levels of exposure. Now, how do we do this in the template model? Now, how do you use the knowledge that we have from our anchor model? So in the center, there is a cholesterol dynamics in the liver, and that dynamics affects the gonads, as I said. And we know that cholesterol is transported into the gonads from the liver. The same is with digestion, the same is with peripheral cholesterol utilization. And we can now use our dose response curve of saying, if this is the normal rate at which cholesterol goes from the liver and the gonads, and you're exposed to dioxin, then this process is changed by this amount. So in this case, we have a dose response curve. In other cases, we could actually have a table. So we can take our anchor model or anchor models and study input-output relationships for all pertinent situations, let's say with diet and with exposure, and compute through simulation input-output relationships in terms of data, and use these tables to inform the changes in the template model. So what we are doing is we are dissecting the big problem into many small problems. Namely, we analyze the anchors, analyze them enough so that we understand what the input-output relationships are. And then we can put everything in the dynamic model for the template and see how the effect overall is on the system. Here is something that is not too important for the understanding of the previous, but I thought it was kind of cool. Because if we look at the template models and the anchor models, the structure of them, they're sort of like dual graphs, dual line graphs. And dual line graphs, if you know about that, the edges and the vertices are interchanged in dual graphs. So in the template model, the variables are actually processes. In the anchor models, the variables are quantities, like the concentration of cholesterol, cholesterol. So there is this funny duality between these two models, which is very important because it tells you when you set up your models how to organize the template model and how to organize the anchor models. So for instance, here we have your cholesterol storage, diet, and so on. And what is handed over from one box to another, from one process to another, is either the concentration of cholesterol or it is a signal that inhibits that process or activates that process. So template anchor models offer a real alternative to other multiscale approaches. We can analyze the anchors individually and then retain just the important input-output information. That information is being handed over to the template model for an overall assessment. The approach offers a clear structure for setting up models. And that structure is very flexible because we can interchange the anchor models. We can just keep them as constant. We can look into these anchor models. We can blow them up and look at more and more detail. In this dioxin case, we can actually show where this discrepancy is not a true discrepancy, namely that you get fatty liver disease, although the biosynthesis is reduced, and we can show why that is. Thank you for your attention.
Thank you for the nice talk. Well, I know there is a coffee break, but we should leave at least one question.
Yes. Typically in multi-scale systems, especially like with fast times, what people do is like do averaging of the fast times here, right? Would that be possible here for anchor systems? Because at the end, it's the slow time scale that determines the dynamics, right?
It is totally up to you. You could average over different things, or you can keep them separate. That's the nice thing. So you have the big model, and you can determine where you want to zoom into. And the zoom, it's not a unique representation of anchors and templates. It's up to you how you set it up. And the variables in template model, they may be anchors or they may be regular variables. Yeah, it's very flexible.
Okay. Can I also?
Sure.
It's not clear to me if it is just a formalism, this anchor and template model, or if the dynamic of the system is actually different from a reaction-based model with the same parameters.
So the way I presented it, it's a conceptual framework for setting it up. And you can set it up as an agent-based model or as a discrete model. You can do it as a pitch-ring model if you wanted to.
So the outcome will be exactly the same. It's just the discrete.
Yes. Yes.